Okay, good. I can see that you can hear me. So last week we started looking at microbiological assays, and we looked at some basics and reminded ourselves of various aspects of cultivating microorganisms. So today we proceed from where we left, and we start off with the handling and storage of microorganisms. So to store them, they are normally stored in culture media, and most often we use agar. So agar is a product from the seaweed. It's solid at room temperature, but melts at around 45 degrees centigrade, forming a gel. So about 1.2% of agar is used to make the gel. We have two different types. So we can have it in petri dishes, or we can have it solidify in sort of a test tube. So when it's stored like this, it's called a slant, and the other is just a plain storage. So this agar can be stored in either petri dishes or as ropes. There are other specialty types like the Rook's bottles or Carrel flasks. So depending on the quantities that need to be stored. For liquid media, they are stored in test tubes that are stoppered. So just a test tube with a stopper at the top. So for short periods, bacteria can be stored in petri dishes because the agar tends to dry out. Slants can be stored at 4 degrees centigrade for several weeks. For storage longer than the 4-week period, then they can be stored at minus 70 degrees centigrade. And in this case, a cytoprotectant such as glycerol, or they may be added, or they can be freeze-dried or lyophilized. So if you have any question, it's better if you just unmute your microphone and speak out. I may not be able to monitor on the chat whether you ask a question. So just unmute and ask the question. You can interrupt at any point in time. So we have seen these different types of media and how they can be used to handle the various aspects. So the key ones are the salants and the petri dishes. They're the same ones that we use to culture. So this is dealing with the transferring microorganism from one culture to another. And we can say that The main goal for surface inoculation, like on this solid agar, is to produce a pure culture. So this is a culture that is descending maybe from one microorganism. So it has similar characteristics or similar behaviors. So this would enable the use of that colony that results from one microorganism for tests of whatever nature that may be desired. Because if you have a mixed colony, then the different microorganisms will behave differently, and therefore results may not be very clear, or you may not get a clear-cut result. So for transfer to the solid agar on a petri dish, but the agar needs to be a bit dry. So it's dried in an incubator until the surface is just slightly wrinkled. So this ensures that when the culture is introduced, then there is no easy flowing. Okay. So it should be a solid surface, and this would also prevent migration of bacteria once they are incubated. To transfer the microorganism, there are two types of implements that can be used. You can use a loop. So a loop, if you look at this one, you can see it's like a needle with something round at the end. So it's just a wire which has been twisted into a loop. And this is used to transfer from the liquid media onto the solid agar. So this can be made from platinum wire or nichrome wire, and incorporate or has a handle for easy holding. Okay, so this is a handle there. Before the transfer is made, this loop is heated in a flame. It can be heated in any type of flame until it is red hot. Then it is put aside to cool down. After cooling down, it is simply dipped into the bacterial suspension in the liquid media, and then drawn across the surface of the agar in these different shapes. So if you look at this slide, you can see some lines drawn in one direction, then in another direction, and then in a third direction. So this would be the first direction or the first strokes that are made. And then this follows without dipping the loop into the media or into the bacterial suspension, such that whatever is being spread on the second stroke is simply what was placed during the first strokes. And the third strokes also, the loop is not reinserted into the bacterial suspension. So in other words, the attempt is to spread the bacteria in single units, such that you'll have a colony forming that is a descendant of only a single unit. So after the spreading or after the streaking, that's actually called streaking. So after that streaking process, then it is covered and the petri dish incubated in an inverted position. So this would prevent any condensate on the lid falling back onto the egg. So incubation is done upside down. Okay. Hope we are still together. So the next this slide now talks about inoculating the slant, and it follows almost a similar process. So, but in this case, the tube would be a narrow one, and you may not get those many strikes that you get. For this one, it's also possible to check for gas production, and all we require is to have the slant either not very vertical, okay? So you can have the slant moving, slanting from the bottom of the tube. Like this one is less slanting. So it is possible to inject the bacteria using a needle rather than a loop inside the egg, such that if there is any bacteria that is producing gas, then it is possible, and that would be an anaerobic type of organism. So the procedure is similar, and eventually this one is covered, but the cover, the cork should not be so tight as to prevent air from going in. So you need to loosen the cap a bit to ensure that air is available for the microorganism. So this final bit is talking about gas production or test for gas production. So a shallower slope or slant is used. For transfer of liquids, we can use the different tools, and when specific amounts are required, then we can use pipettes which are able to take the desired volume. So you have used a pipette before, and maybe you have tried to use your mouth to pull the liquid. So this is unacceptable. So you should one should only transfer using the automatic pipette or one with a pipette filler. So this is simply a bulb, an elastic bulb. But once you press it, you're able to draw some liquid as you release it. Also, to prevent the solution going up into the bulb, then you can have some sterile cotton wool coming in at the top of the pipette. So it's important to learn about different ways in which transfer of microorganisms may occur unintentionally. So, and these are the two types of ways in which transfer may occur, and it's just dependent on the size of the droplet that is formed. So there are those large types of droplets greater than 5 microns. So these are likely to contain very many microorganisms, and because of their size, they do not move very far from the area of operation. So these ones will contaminate immediate environment and working surfaces. So the person handling or in that area can get the infection or even now pick from there and transfer. So the other one is the smaller droplet, which is less than 5 microns. And these ones will dry almost immediately, leaving behind the microorganisms suspended in air. So this one can then travel depending on the conditions of the environment. Is it windy? Is it an open area? So these ones will remain suspended in the air and can travel to further distances. So you may get somebody who is not even in that building getting the infection. So, and this one would tend to cause lung infections. So how are these droplets produced? There is splashing, and this would occur maybe during transfer of a liquid from one tube to another, or trying to mix the suspension, the bacterial suspension, using the loop. So as that happens, then you may find some droplets forming. In the other instances, when the loops are being heated, if they have been dipped into this suspension and you heat them, that heat may evaporate the liquid while dispersing the microorganism in those small droplets. So it is possible to form those droplets during the sterilization period before the microorganism has been killed. In the other scenario is where the hot loop is dipped in the full liquid suspension, and because of that heat, the liquid tends to evaporate in those fine droplets, carrying away the microorganism with it. Right? So anaerobes are those microorganisms that we said do not require oxygen. So, and it is a bit difficult to cultivate them. However, there are tools which facilitate that cultivation. So you must find there must be a way to remove any oxygen that is present. And this is achieved by use of thioglycolate. So it's an additive to the medium. Which removes any oxygen present. An indicator to show that oxygen is clearly removed is methylene blue. This is a redox indicator. But in case the indicator is toxic to the microorganism, then it can still be removed. So anaerobic jars, so the tool that is used known as anaerobic jars, they are mostly made of clear polycarbonate. You can see this one, though it may not be very clear. So that is made of polycarbonate, and it has a lid. This is a mesh. So it's a container which is a mesh, so it's open. It has holes in it, and that container holds a catalyst made of palladium. So this palladium is able to catalyze the use of oxygen by whatever chemical is there. And usually it produces hydrogen gas, which reacts with the oxygen, catalyzed by the cold palladium catalyst. So any oxygen is used up to form water. In some cases, some microorganisms do well when there is carbon dioxide added. So this that carbon dioxide would still be produced by that package. So this is just an example of a typical anaerobic cultivation kit. So this is called a gas pack. There could be other brands. Some types of media may also be used for cultivation of anaerobes, like Robertson cooked meat broth. Robertson cooked meat broth, which contains 5% cooked meat in nutrient broth. So hemine and glutathione present in the meat act as reducing agents. So they react with the oxygen and remove it from the environment. For agar plates, like petri dishes, then anaerobic condition can be maintained by covering the surface with oil. So there are different ways in which the anaerobic condition may be achieved. Either the chemical route, where the hydrogen produced by the package reacts with oxygen, or the use of the cooked meat, or covering agar surfaces with oil. So in most of the microbiological assays, the goal is to check on whether there is bacterial growth and how much. Sometimes you may want to quantify how much growth has occurred. And one of the ways to do that is to count the actual number of microorganisms present. So to do this, there is the manual way of doing it. And one of those is by use of a counting chamber. So this uses microscopy with a unique slide that allows for counting the microorganisms. So this slide is a hemocytometer counting chamber. And 1 square millimeter is divided into 400 small squares. So these are only visible under the microscope. And each of these 400 squares each has a depth of 0.1 millimeter. So you can calculate the volume of that one small square. So it is possible to count the number of microorganisms in that small volume and then determine how much you had in the total volume. So the normal microscopy, the transmitting light microscope, may not be able to observe the microorganism because of the nature of the microorganism. We said it's about 80% water. So various methods, various microscopes are used to count live microorganisms. But in most cases, the bacteria may need to be killed or fixed. So multibacteria may need to be fixed, and this is achieved by adding 2 to 3 drops per 10 ml, 2 to 3 drops of formaldehyde, 40%, to a 10 ml suspension of bacteria. So other ways, the microorganisms may be added onto the slide as a drop, allowed to dry, and then an eyepiece that has those small squares etched onto them used to count the microorganisms. So in the second case, the eyepiece is the one that has the squares. So it's just allowing you to map the area on the slide and then count the microorganisms on the dry slide. So other ways in which the microorganism counting can be achieved is by use of spectroscopic methods. So these ones are easy to use. They are rapid, so they can count many slides. However, they require careful calibration, so they require expertise. These spectroscopic methods may rely on the use of colorimeters, nephelometers, or spectrophotometers. So these are the different principles that may be used to count the microorganisms. So spectrophotometers seem to be superior to the other two. An important thing to note is that dilute solutions of the microorganism suspension should be dilute to improve accuracy. The third way in which counting may be done is by use of electronic applications. And these are simply counters that have been made to be able to respond the presence of a particle. There are all those different ways in which that is achieved. Microcalorimetry. We can measure the change in resistance or conductivity of the solution. We can use radiometric or infrared to monitor carbon dioxide production. Then there are the other methods. You can measure the total biomass, total nitrogen, dry weight, etc. So all those are different ways in which growth can be checked. The growth of a microorganism can be checked. So the methods that we have outlined for counting, we are saying that the bacteria need to be fixed. So you cannot really tell whether the bacteria died during fixing or it was dead. So in cases where you need to determine the number or the proportion of microorganisms which are alive, then you do variable counts. So variable counts, you want to count only the cells that can divide. That means they are living. So when reporting, you'd see that these counts would be reported as the number of colony forming units, because each of the microorganism, each specific cell is able to produce a colony. So each is reported as a colony forming unit. And this would be per unit volume, like ml. So the technique involves diluting a known volume of the bacterial suspension and then spreading a specific volume to an over-dried agar plate. So this can be evenly spread using a sterile wire or glass capillary. And because the agar is slightly over-dried, the liquid will soak into the plates, leaving behind the microorganisms on the surface. The agar plate is then inverted and incubated. So this is done with several dilutions and any number of replicates required to achieve colonies made of about 30 to 300 per plate. So the target is to have like 30 to 300 colonies of viable microorganisms per plate. So these are other ways in which viable counts can be done. There are power plates, membrane filtration, and ATP determination. So all these are different methods that can be used to determine the number of microorganisms which are living. So as we mentioned, there are many different types of microorganisms, and it is easy to have contamination between or with any culture. So it's important that we have a system to identify what microorganism we are dealing with, what are its characteristics. So this would be a necessary step, especially when dealing with microorganisms from environment, from an infection. Maybe you want to test whether the antibacterial agent is effective on the microorganisms causing an infection. So it would be important to be able to classify and identify. So this would only be done if you have a pure colony. And this, as we mentioned, is achieved by streaking the microorganism suspension on an agar plate, okay, and incubating. So that way you are able to see the behavior of the colonies. In other cases, selective enrichment media may be used. So this is type of media that would encourage the growth of the one microorganism while preventing the growth of other microorganisms. So this is an enrichment broth type of media. So generally, there are many types of microorganisms, about 500 genera. And to identify them, we use the binomial nomenclature. It would be difficult to call them by common names. So to get to the real identity, you may need to use genetic relationships. So you need to do genetic typing, which is not very easy. So the most accurate indicator of a species would be defining its gene or genetic makeup. Under normal circumstances, simpler tests and routines can be used to check for phenotype. And if done in a logical and sequential manner, it is possible to identify or at least come close to the identity of the microorganism. So the kind of phenotypes that we would be looking at would be the morphology. How is the cell? How is the microorganism? What is the shape? Then we would also look at the staining characteristics using the different types of stains. Then move to cultural reactions. So cultural would be what is the shape of the colony, or is it forming a colony? What is the shape? What is the size? What is the color? What is the texture? Is it rough, smooth? Is it producing any odor, the smell? Is it colored? Okay. And then is growth in aerated areas or in anaerobic areas? Okay. What is the temperature, the optimum temperature for growth of the microorganism? And what is the optimum pH for the microorganism? So this would be cultural related properties or phenotypes that would be important for identification. Then we have biochemical reactions. Those some microorganisms have unique abilities to carry out some enzymatic activities. So we can use this as markers for identity. So closely related bacteria may be told apart by using biochemical reactions. So this may be done in the old ways or using kits. So old ways is putting in the different mixtures there and then testing for the reaction, or using kits specifically made for one purpose. Okay. So classification and identification, we use morphology. Okay, morphology being the cell size, the shape, the formation of spores, aggregation, and whether they are motile, staining, whether they are gram positive, gram negative, whether they are acid fast, or they have spores. So there is a spore stain that can be used for that. Cultural reactions, you have mentioned the culture, the colony formation, the shape, the size, the color, texture, smell, pigments, and etc. So this one I have left it for you. I want you to check for fungi along similar lines that we have used for bacteria. How are they cultured? The media that is used? What is the nature of their colony? How are they identified? Etc. So you check on that as your assignment. Any questions? Okay, so we now look at specific applications and how we use the microorganisms for various pharmaceutical applications. And this is the outline of what we'll discuss. Which factors need to be controlled? Then we'll look at antibiotic assays. This is quantifying the potency of an antibiotic. We will look at how we can determine minimum inhibitory concentration. Then we will look at preservative efficacy tests, which are also known as charge tests. And then we will see how we can evaluate the effectiveness of disinfection. Then we'll also consider the quality of pharmaceutical materials, the microbiological quality of pharmaceutical materials, and these include non-sterile products and sterile products. So measurement of antimicrobial activity. So there are two ways in which these are performed, but two important things need to be noted. Standardization, which we mentioned, that we must use standards, and then we must control factors. So there are various factors that need to be controlled for the result to be acceptable. So the two ways in which these are done, the first one is the inoculum of test organisms. So this is the bacterial or the microorganism suspension is added to a solution of the test chemical. So this would be the analyte. Then incubated. Samples would then be removed over a period, and the growth or inhibition of growth determined over a period of time. At the end of the run, then the test chemical is inactivated, and proportion of survivors can be determined. So in this case, you'd have a negative control that would be giving you the 100%, and then you calculate the proportion, those two proportions. So this is the most common way in which antimicrobial activity is done. The second way is where you have the culture medium and the chemical being present, and then the degree of inhibition of growth is determined. So we'll see both in slightly more detail. So which factors need to be controlled? One of the factors that need to be controlled is the origin of the test organism. Organisms that are microorganisms which are in the same species may show different behaviors or different phenotypes. So these organisms are said to belong to different strains. Okay? They belong to different strains. So it is important that any test, any microbiological assay that is done should specify the strain that was used in the test. This would allow reproducibility or enhance reproducibility of the assay. So the source of the microorganism is important because it can influence some of the properties that will affect the measured activity. And we have an example of the different sources of the microorganisms. So we have human or animal infections. So this would be maybe a specimen acquired from a patient in the hospital or a sick animal. So this kind of microorganism or this kind of microorganism source tends to have to produce colonies that are more resistant to antimicrobials, especially antibiotics. So these are microorganisms which are growing in an environment where there could be some low levels of antimicrobials. And therefore, they have learned to live with those antimicrobial antibiotics. So this would show some poor response to antimicrobial being tested. So you'd report that the microorganism has a lower response compared to microorganisms from other sources or that are not exposed to the antibiotics. The other source, maybe you are testing microorganisms which have contaminated some medicine batch. So this would be more resistant to chemicals used as preservatives, okay? Because they have been exposed to preservatives in the product, and therefore they are actually growing in the presence of those preservatives. So the source is very critical. It's important to report on the source of the microorganism used in the assay. In addition, official methods, or what we call monographs, limit the number of times the culture specimen may be grown in fresh medium. So this is noted as the maximum number of subcultures or passages that are allowed. So because culturing in fresh medium, as we noted, the bacteria or the microorganism can change its behavior over time. We recall the rough and smooth colonies that we mentioned. So official methods will state that if a culture has been subcultured several times, then it is not appropriate for further use as a test or a standard. The other factor that needs to be controlled is the composition and pH of culture media. So it's important to know that some culture media may naturally contain some components that can affect the growth of microorganisms, either positively or negatively. And some mixtures are known to have substances that will antagonize the activity of certain antimicrobial agents. And for this, we have in mind para-amino benzoic acid, for example, or thymidine, which tend to antagonize the activity of sulfonamides. So if you remember the mechanism of action of sulfonamides, then it makes logic, or it is logical that PABA will antagonize the activity, or you'll see poor activity of the microbe, the antibacterial agent if you are testing a sulfonamide in the presence of PABA. You can also have divalent or trivalent cations, which would antagonize the activity of tetracyclines. So they would form complexes with these tetracyclines, and then you don't get the active form of the tetracycline. So you'd report that as low activity. These cations also antagonize aminoglycosides in a similar way. Then, as we mentioned when we were talking about the nature of the bacteria, we say that bacteria membranes act as semi-permeable membranes. So they allow some molecules to go through while preventing others. So being a bilayer, the bacteria membrane is permeable to drugs which have some lipophilicity or liposolubility. pH can affect the nature of the drug molecule, especially for weak acids or weak bases. So depending on pH, you may find the molecule as ionized or unionized. The unionized form is the one that would be able to pass through this bacterial membrane. So ionization, the pH and pKa of the drug, the relationship between those two is critical. So if the pH promotes the pH of the media promotes the non-ionized form, then you would have more permeation through the membrane, and therefore more potency would be observed. And vice versa, if the pH is promoting the ionized form, then you would have low permeation through the membrane and therefore low activity. For example, benzoic acid, which is used as a preservative in oral mixtures, is more effective in acidic buffers. So benzoic acid is a weak acid, so raw pH would promote the non-ionized form and therefore more penetration. Weak bases such as aminoglycosides, the streptomycin, gentamicin, neomycin, are slightly more potent in alkaline media. Then we have presence or absence of organic matter, such as blood, pus, serum. So these ones tend to reduce the potency of the antibacterial agent, measured potency of the antibacterial agent. So these ones tend to be protective on the microorganism. The other factor that need to be controlled is exposure and incubation conditions. Um, And these conditions include temperature, which is an obvious reason. Then there is duration and redox conditions. So these conditions or these factors need to be controlled even if reculturing is being done to test survival. So usually when we are testing survival, the microorganism will be removed and placed in fresh media. So those conditions, the conditions that the new culture is being established in is important because it could either promote or inhibit the recovery of the microorganism and eventual growth. So for temperature, we'll note that increasing the temperature of exposure of the test organism tends to increase antimicrobial activity. The extent to which temperature enhances activity is measured by a temperature coefficient known as Q subscript 10 value. The Q10 value so Q10 value. So this is the number of times the activity of an antibacterial or antibiotic agent increases for each 10-degree centigrade rise in temperature. So this value is different for different types of compounds. For example, phenols have a Q10 of 3 to 5. Alcohols have a Q10 of greater than 10. So that's the number of times of activity increment by changing the temperature alone. So most pharmacopoeia allow a 5-degree centigrade variation. So you can run, if they say you're doing the tests at 37 degrees centigrade, they allow from 35 to maybe 40. So you can see the effect of choosing a different temperature to do the tests. So it's important that that temperature that is used is actually controlled. In terms of duration, as the microorganisms that don't die very quickly after introduction, some of them will adapt in the new environment and start growing within no time. So duration is very critical because if you have some resistant microorganisms, even a very small percentage, and you are incubating for a long period, then it means those resistant microorganisms have enough time to recover and multiply. So like in this slide we have, this chart we have here, you can see that you have 100% at time 0. So when it is introduced into the chemical, the value or the numbers decrease within 6 hours to around 0.01%, which is a very small proportion compared to the beginning. But then within 48 hours, you have the number of microorganisms increasing to 100% once more. So within 48 hours, or within 42 hours, whatever remained 0.01 has multiplied and replenished the initial number. So it's very critical that duration of the test is maintained. So the organism may adapt and become resistant to the chemical, or it was initially, some of them were initially resistant and survived the first few hours and multiplied. So in preservative efficacy tests, exposure period is 28 days. So this is enough time for any microorganism, any survivor, during the initial 24 to 48 hours to recover and divide. The final population may even be higher than the population beginning, but this would have different characteristics from the initial profile. So if you are to measure maybe antibiotic activity, antibiotic responsiveness of this population and compare it with this population, you would get different values. So this chart was done from Pseudomonas aeruginosa. This is a response to benzethenium chloride. Okay. So you can see the response there with time. So similar observations may be seen where you are testing for minimum inhibitory concentrations. The other factor that we have mentioned is the redox condition. So in low oxygen concentrations, some antibiotics like aminoglycosides are far less active, while metronidazole is more active. So we know one of the mechanisms of metronidazole is by increasing oxidative stress. So in presence of or in the absence of oxygen, then metronidazole would be more effective. On the other hand, if you're doing agar diffusion assay as a method, you see what agar diffusion is. At the surface of the agar, you might have a larger zone of inhibition compared to the bottom. And this is simply because oxygen is not effectively diffusing through the agar layer to reach the bottom zone. And therefore, the response of the microorganism at the different zones will be different. So redox conditions is also an important factor to control. The other factor that is critical is inoculum concentration and physiological state. So if you look at inoculum concentration, if you are putting a fixed volume of the inoculum, and then you have different numbers of viable microorganisms, where you have more microorganisms to start with, then you'll have a higher probability of slightly more survivors. So in this case, then growth would be very vibrant if you were to do survival rate. So if you use high inoculum numbers, many microorganisms added to the test solution, then what you will be noticed is that the antimicrobial agent will be less potent. But this simply because a higher number of microorganisms were introduced from the beginning, and therefore chances of survival increased for the microorganisms. The other factor is the history of inoculum or the physiological state. So as you recall, we said that the microorganism as or the colony undergoes different stages. So immediately after introduction, the microorganism is adapting to the new environment. After that, there is exponential growth. Then there is a stationary phase where the death and multiplication are almost the same. And then there is decline. So depending on when the inoculum is picked for incorporation into the test, it is going to influence the measured activity. For example, if you measure when the microorganisms are already dying, then these are not very vibrant in terms of responding or adapting. So you are likely to measure high potency. Unlike when you take microorganisms which are actively dividing, this is a young population and therefore more adaptable. So the history of the inoculum or the physiological state of the cells, the microorganisms that are added into the culture for testing need to be clearly defined. And also to add to that is that bacteria are known to synthesize inactivating enzymes like the beta lactamase. So if you have a large number that you add to the culture, so you are increasing the production of these inactivating enzymes, and therefore more of the drug molecules will be inactivated, resulting in measured low activity. Additionally, as you increase the volumes of the inoculum that you're transferring, you're also transferring carryovers. So if the beta lactamase was produced in the previous culture, you'll also be carrying that in solution and including it in your test. So that carryover effect may also be observed on your result. Okay. So any questions so far? No question. Okay, so let's look at antibiotic acids. So this is just an introductory slide, and it's just saying that there are three main areas or main ways in which we analyze antibiotics. So the first one is the conventional chemical or instrumental. So this is titration, spectrophotometry, HPLC. So that is one way in which some of the antibiotics can be assayed. Then we have enzyme-based and immunoassays. So this we are going to look at in due course. And finally, the biological assays or the microbiological assays that we are looking at now. So each of these techniques has its strength, its applicabilities, and preferences. For example, enzyme-based and immunoassay kits are more likely to be used in a hospital setup for therapeutic monitoring of toxic antibiotics like aminoglycosides and vancomycin. While in the manufacturing, they will try to use the HPLC for quality assurance applications. So, of course, wherever possible, majority of the people will select conventional or chemical methods because of ease of application. So biological assays are mostly used when alternatives are inappropriate. Okay, mostly used when the alternatives are not very appropriate, and we have given a few of the scenarios where biological assays are the most applicable ones. If it is difficult to separate the active in the active compound from whatever it is, then biological assay would be appropriate. If the degradation products are interfering, or in cases where derivatization may be required, maybe to enhance absorption of UV visible for detection. So we have given that example of aminoglycosides. So let's expand those by giving specific cases. If the solution where the antibiotic is has many complex substances which can interfere with the chemical assays. And in this, we have an example of fermentation broth or a biological sample like serum or urine. So this may contain many substances which are capable of interfering with chemical assays. Maybe too much background noise such that it is not possible to carry out a chemical assay. In another case, if you have or you expect that the antibiotic has been degraded to a large extent, and therefore you have very many related breakdown products in the sample, then biological assays are indicated, and a specific scenario is during drug stability studies as part of product development. So as when they're determining the half-life or the shelf life, they need to carry out accelerated degradation tests. So you'd expect that a good number or a good proportion of the drug will be degraded under those circumstances. So the best route to test for activity or potency then would be a biological assay. In another case where this method would be appropriate is whereby the sample is being extracted from a formulation. So you already have a product in the market. You want to see whether, or you have a final product. You want to see to test the potency of that product. So that product is going to contain other substances added during the manufacture. So some of these excipients may interfere with chemical tests. In another case, for example, you may have products that in the market they are mixtures. They are mixtures of related compounds, either as isomers or compounds which are maybe produced by the microorganism during production at the same time. So in this case, we have an example. We have an example of an antibiotic like neomycin. So we have this neomycin, and depending on the substituent there, then you have different compounds. Okay? So each may have different potencies, and therefore a biological assay would be able to show the activity, because it's not possible to determine this by chemical means. So the commercially available products, it's important to monitor from batch to batch, because this proportion of active ingredient may vary. So that is a good reason why microbiological assay would be used for such a case. So the types of antibiotic assays, there are two main methods. We have what are known as agar diffusion and turbidimetric assays. So these methods are clearly described in one of the pharmacopoeia, the European pharmacopoeia. You check 2004 edition, you'll see the details, the experimental details or the test details, such as the test organisms, the solvents, the buffers, culture media, and incubation conditions. Okay? So these are clearly outlined in the pharmacopoeia. So from the beginning of production of antibiotics, antibiotics were mainly unavailable in pure form, and therefore chemical methods were rarely applicable. So when assaying for the content, potency or activity was expressed in some units known as international units of activity. And some of the antibiotics are still reported or doses or dosed in terms of these international units. I'm sure you've seen some of this in your clinical area. So nystatin, I'm sure you have met it. Neonatal drops, for example, polymyxin. So these are usually dosed in terms of international units. So most of the other antibiotics will be dosed in terms of micrograms, or the formulation is going to report in terms of micrograms per ml, etc. So antibiotic acids are used in form of a potency ratio, which is the activity of the unknown relative to the activity of a standard. So what is agar diffusion? So agar diffusion uses the solid media, which can be done on a petri dish or specific plates made for antibiotic acids. So the petri dishes may not hold very many microorganisms, so they are limited in terms of the number of tests that can be done at the same time. We'll see a plate, an example of a plate that is used for this test. So what does it involve? It includes the agar. So this is a petri dish cut in half to show the agar filling the petri dish, to show the depth. So this is the depth of the agar. And this is a hole cut out in the solid media. Okay. And this hole is where the test or the standard solution is going to be added. So the drug, the antibiotic, will diffuse through the medium. Okay. And at a certain point, the concentration will be high enough to prevent the growth of the microorganism which is on the surface. So this covered area, or this is growth has taken place, microorganism multiplication has occurred, but this clear zone here, the microorganism has been prevented from growing. So the diameter of this circular zone is dependent on the potency of the antibiotic that is added. Okay. So the tools which are needed are the plate, the agar, of course, the loop for adding the test organism. You'll also need a cork borer, which you can use to drill that hole, and the solutions of the test and the standard, the incubator as well. So this area of inhibition, or the zone of inhibition, will depend on potency of the drug, the ability of that molecule to diffuse through the agar. Water solubility is also a factor that can influence whether the antibiotic can be tested by this method, as well as the charge. Is the drug ionized or not? So there are some very effective antibacterial agents that cannot be tested by this method, like the parabens, because they will not diffuse from the well. So this is an example of an agar diffusion assay plate with showing clear zones of inhibition. Okay. So this would be the standard drug, and this would be the test. Okay. So you can note that the principle of agar diffusion is that the chemical diffuses from the well, creating a concentration gradient. At some point, the concentration is high enough to prevent microorganism growth. The edge, or the clear zone of inhibition, the edge of the clear zone of inhibition indicates the critical inhibitory concentration. If I go back a bit here, so at this point, this is where you have the critical inhibitory concentration. Beyond that, the concentration is too low to prevent growth. Inside of that, then the concentration is high enough to prevent growth. So this is different from minimum inhibitory concentration. In agar diffusion, we have critical inhibitory concentration. So the zone around the well is clear, the rest is opaque due to microbial growth. The diameter of inhibition will depend on the concentration of the drug in the well. So what is the output of this test? The diameter of the zone is measured, and this can be measured directly using calipers or by projecting on a screen and then measuring. So this projecting on a screen and measuring on the screen will magnify that and therefore enhance or increase the accuracy and precision of measurement. You can also know that the placement of the tests and the controls is randomized. So this ensures that differences or changes that may be observed or natural, maybe the depth of the agar is not uniform. So the result would be randomized, and therefore such errors would be accounted for by that. So the pattern that they normally use is what is known as the Latin square method for randomizing placements. So this is a typical result. So if you are to plot the zone of inhibition, the diameter, against concentration of the antibacterial agent, if the concentration range is not very wide, then you would tend to get a straight line. So if you plotted diameter against concentration, you get a straight line. So if this from 0 to the highest is not very wide, you get a straight line. But if you measure over a wider concentration range, then you get a curve sort of. So it's not straight, but you can get a straight relationship if you square the diameter. So this is the same measurement, so plotted against the zone diameter, and the upper is plotted against the square of diameter. So squaring the diameter and plotting against concentration, you tend to get a straight linear relationship. So the usual test is to run the antibiotic standard in 2 or 3 concentrations and the test in 2 or 3 concentrations in a similar way. So of course, the usual duplication is required. Okay. So if you use 2 doses, it can be determined, the potency can be determined either from a plot of the zone, the plot of the zone and concentration, or using an equation. Okay. I don't know whether yours is showing some equation, but what we have is log x is equal to LDR multiplied by U let me redo that. Okay. Let me call this high this is UH. It's not working very well. UH plus UL minus. So here define these terms SH plus SL. So this divided by SH minus SL plus UH minus U. This is minus UL. Okay, so that is an equation that you can use to calculate the relative potency. LDL refers, so X, log X, or X is the potency ratio. So log X is the ratio, the log potency ratio. LDR is the logarithm of the dose ratio. So you, it means you have a dose, UH, and you have a dose, U high. So the low dose and the actually not the U but the S. So it is SL per S high. So this is the low concentration of the standard dose, and this is the high concentration of the standard. When you do that ratio, that is the dose ratio. So LDL is the log, the logarithm of that dose ratio. UH and UL refer to the concentration of the test or the unknown, the high concentration and the low concentration respectively. SH and SL are the high and lower concentrations of the standard. So, and these values are the zone diameters as measured. So SH, SL refer to the zone diameters due to the high concentration of the standard and the low concentration of the standard, respectively. Similarly for UH and UL. So, and this is a representation of this equation. So this one has been derived and it's known to work, but you can still do it manually using this kind of plotting. Okay. I see it's 11. Are you having another class? Michael? Michael, you there? Anybody? Do you have another class?
Hello, yes, we have another class at 11.
Okay. So let me wrap it up here. So you can check out this chart and look at the way you can use it to calculate the potency of a drug by that method. So we meet again tomorrow by same means for chromatography. Thank you and have a good day. If you have any questions, you can write them to my email or talk to me.